Thank you for that great introduction. Today, I forgot non-summer dynamics. So in limnology, as an undergraduate student, statement said the definition of limnology is the study of lakes located within 60 miles of a major university campus, but not during the winter. And so unfortunately, this tends to be true of most of our research and kind of governmental monitoring programs. And so the question remains, what are we missing with this summer-centric focus? What is the sound of winter? We're seeing increasing incidences of harmful algal blooms occurring more times, more places, more often. We're also seeing declines in our oxygen concentrations, particularly in the bottom of our lakes. And this, of course, has implication for things that like to breathe, such as fish and fish habitat, which we can see some negative impacts resulting from. So questions remain is how important is winter? And are changes that are happening in winter setting the stage for some of these summer conditions we're starting to see more of, specifically looking at these harmful algal blooms, as the green shown in the picture here. So this is one of the most famous images in limnology now. This is an algal bloom in the western basin of Lake Erie. Satellite imagery, so it's literally, the bloom is so large that it can be seen from space, and it's the green color shown in the image. And this is not just a Lake Erie problem. We're seeing these extreme algal blooms expanding on a worldwide basis. And so there's something that's happening on a global basis that's changing or making life better if you're a cyanobacteria or someone who tends to bloom. And when we go into this, we're set out with these traditional paradigms of what to expect of algal biomass in lakes. And so here on the x-axis, we've got a year-round data set from January through to December. And for a lot of this presentation, I'll be referring to chlorophyll A concentrations as a proxy for phytoplankton biomass. So whenever I show chlorophyll, you can kind of think of that as being the number, the amount of phytoplankton that's there. And so typically we see this bimodal pattern where we have the peak concentrations happening in the spring and the fall. And this is, kind of the basis for a lot of our monitoring programs. And so typically within those green lines here in the summer is kind of the classical sampling season for most programs. And so we've had this traditional summer-centric lens where all the good things or all the big changes are during the summer months. And there's not as much focus on the shoulder seasons. So what we're headed into now, the fall and the spring, and then of course the winter. But we are seeing more incidences of winter algal biomass. And so this is an image at the surface is ice cover, which is very clear. And what you're seeing is the green is an algal bloom under the ice. So this is becoming a lot more common now, of course, looking for it. There's important implications for what's happening around the world these days, which is the winters are getting shorter. And so we're seeing changes in this unmonitored season. So it's important more now than ever to get that baseline information as to what everyone's doing under the ice because things are changing. Unfortunately, if you're a pessimist, you'll come to the realization that the percentage of ice-free winters will increase from 2 to 60% by the end of the current century. And so we're already seeing evidence of that here in North America, and certainly in Europe, they've already seen lakes completely lose their ice cover. And this is being found, as I said, on a global basis. So this is a summary paper, Hampton et al. In Ecology Letters a few years ago, where they compared the chlorophyll A concentrations of ice in the winter to those in the summer in the same lakes all around the world, and found that the under ice chlorophyll A concentrations, so here in the box plot, are about half of summer. And so, you might think, well, this is what we'd expect, but, according to our textbooks and kind of these traditional assumptions, we probably expect it to be pretty negligible. So I would argue that half is an important number and one that should be, considered in models and these kind of perspectives on year-round chlorophyll A concentrations. So I'd suggest that the take-home message here is that those winter chlorophyll A concentrations are not zero, and half of summer is not a negligible amount to ignore. So the objective of today's seminar is to kind of ask the question, what happens in lakes in the winter? And then more specifically, do we see that feedback loop with summer conditions, and does it matter? And so a lot of our assumptions as to what's going on down there in the winter is that nothing, right? Nothing happens in a fridge. It's too cold and dark, so we don't see a lot of biological mechanisms or biogeochemical transformations occurring. And we also, admit that maybe something's going on. And here's that image of the right combination of light and nutrients. You can get these under ice algal blooms. But due to logistical reasons, it's just too difficult to get out there, and we often don't measure it. Either we're faced with uncertain and unsafe ice conditions, or as I said, with these declines in ice cover, going out in snowmobiles, as shown here in the image, is becoming less possible. And so I want to kind of open up the black box of the winter and shoulder seasons and look at this year-round phenology. So these patterns of chlorophyll A or phytoplankton biomass. Look at year-round cyanotoxin concentrations. So of the chlorophyll A, a portion of that is cyanobacteria. And we think that life is getting better for them and are changing so that they're out competing the other types of phytoplankton and maybe facilitating their production of toxins. And then metabolism, looking at that relationship between primary productivity and respiration under ice. And we can do that across a latitudinal gradient of lakes and reservoirs. So although I realize this is a glacial lake group, I'm going to mix and match in my presentation between man-made reservoirs and natural lakes. Unfortunately, where I am now in the University of Missouri, the state of Missouri only really has reservoirs. So I'd work on the natural lakes that existed here. As I said, I'm just going to kind of mix and match lakes and reservoirs.
Dr. North.
So this is yeah.
We're getting a little bit of garbling on your audio. Do you think it would help if you turned your video off?
Sure, I can try that.
Thank you. Sorry for the interruption.
Yeah, no, thank you for telling me. I would have just kept talking. So this is a conceptual diagram of winter futures. And so we often call our northern lakes or temperate lakes dimictic, meaning they have this ice cover that forms during the winter months. Down where I live here in Missouri, the lakes fall into this warm monomictic category, meaning we rarely get ice cover. And so in the winter, we end up kind of having this circulation pattern down to the bottom of the lake. And then as we move farther south, or as I'm hypothesizing here, farther into the future of our climate change, we can get permanently stratified lakes. And so these are ones that don't mix, and the bottom waters are separated from those top waters. And so we can kind of use a latitudinal gradient of lakes along this continuum, where we see differences in the mixing patterns. So having that ice cover means that the light can induce this convective mixing. So it actually can mean more light for the phytoplankton because they're kind of stuck up in these higher levels. And so here, this dial here is showing how much mean light is available, which is a combination of what's coming in from the sky, incidence irradiance, what's getting through that snow and ice into the water, and then the mixing depth. So these capturing these kind of convective mixing layers. And so there's actually quite a bit of light if you have really clear ice and little snow cover in these dimictic lakes. Moving down the climate continuum to warm monomictic, because now there is no ice cover, that means that the lake is circulating from top to bottom. And so if you have a fairly deep, say 50-meter lake, those algal cells are kind of at the will of the waves. And so they're being dragged down into the bottom of that water column. And then we can go again in the future to these permanently stratified ones where again, there's a separation between the top waters and the bottom waters, and they're able to stay in those top waters. So this kind of sets out the hypothesis for this time for space continuum comparison that I'm about to make. So here is the study systems ranging from some Canadian lakes and reservoirs. So here you can see up in Saskatchewan, the ice duration can be over 200 days in some cases, very long winters in Saskatchewan. In comparison with Lake Simcoe over here in Ontario, which has an ice duration anywhere from 70 to 100 days. And I'm contrasting those with the Missouri reservoirs and a reservoir over here in Kansas, which have very little ice duration, if any at all. I'm also using a range of study systems in terms of trophic status. And so oligotrophic are the squares, and those tend to be really clear blue lakes. And those range to hypereutrophic, mostly down here in Missouri, and one in Saskatchewan, that are the really kind of green colored lakes with lots of nutrients and lots of algae in them. And so when we think about what controls algal growth, we often look at these 4 different factors. So we know phytoplankton are primary producers. They require light in the sun. We also know that water temperature is an important factor. One of the most famous papers in limnology is called Bloom's Like It Hot. So we know that particularly those cyanobacteria, the ones that have the potential to produce toxins, are going to do better under warmer conditions. We have nutrients. This is, the plants are sustained by both nitrogen and phosphorus. And so we need to have that present to control algal growth. And then, of course, the top-down processes like grazing. And here the picture is depicting Dreissenids or zebra mussels, but also it could be zooplankton or anything else. So we can kind of look at these top-down and bottom-up controls and make predictions of how things are going to react. Here's a figure from Lake. So it's shown here, the water treatment intake pipe. So one of the ways internet problems. One of the ways you can get around having to get out in the ice on a year-round basis is to use the intake pipe of a water treatment plant. The Ontario government has been doing this for decades. And it's, some of the most unique data sets we have that show these things on this basis. And so here I'm showing data of phytoplankton biovolume on a year-round basis on Lake Simcoe, back from before there was zebra mussels, so 1982, till the darker colors of the years after the zebra mussels invaded. And in fact, now they're all quagga mussels, but this is some older data I'm showing. And so we can see here that prior to zebra mussel invasion, the clear ones show that kind of bimodal pattern that we would expect at a phytoplankton biomass, with peak concentrations happening in the spring and the fall. But then once the zebra mussels invaded, we saw this change. And in fact, we see a lot lower biomass. And in particular, the highest concentrations are occurring now under the ice in January and February. And so we are seeing some indications that changes in top-down processes could have impact on these year-round dynamics. So that was grazing. Moving on now to nutrients. I did some nutrient status indicators, so a variety of different ways to basically ask the algae, do they have enough nutrients? And specifically looking at phosphorus at the top here and nitrogen in the bottom of these tables. And so on all of these systems, ranging from the Saskatchewan reservoirs, Diefenbaker, Broderick, and Blackstrap in St. Denis to Lake Simcoe in Ontario, we found that whatever way we measured it and however we gave them nitrogen or phosphorus, they always had enough during the winter. So we don't think nutrient deficiency is having a control on phytoplankton biomass in the winter months. We know that a lot of the processes that deliver these high nutrients under ice are related to the low oxygen conditions that it can occur under ice cover. And so as the phytoplankton grow at the top of the lakes, they sink down to the bottom where they're consumed and decomposed by bacteria who don't need light. This is how we get dead zones in lakes, or Gulf hypoxia you might be familiar with. And so when those low oxygen conditions occur, that changes the redox chemistry at the sediment water interface. And phosphorus that used to be kind of tightly bound in the bottom suddenly gets let go and comes up into the water column and becomes available to where the algae are living and growing. And so we call this process internal nutrient loading. And I looked at this specifically on Lake Diefenbaker, which is a reservoir in Saskatchewan, and found that the ratio of internal phosphorus loading between when the reservoir was ice covered to when it was ice free was about 3 times. So the loading rate was higher, 3 times higher in the winter or the ice covered months than it is in the summer. And so this kind of explains, I think, in some ways why the phytoplankton were not phosphorus deficient, because they have this additional supply of nutrients coming in, historical nutrients or legacy phosphorus from the bottom waters and the sediments. So the next, as we said, is temperature and light as indicators of algal growth or controls on it. As I mentioned, they are primary producers, so they do need a lot of light, but how much actually gets through under the ice cover where the algae are living depends on the depth of both the snow and the ice, as well as the type of ice. So some of you might have been out ice fishing and being able to look right down through the lake and watch the fish swim beneath your feet. We tend to call that kind of black ice or clear ice. And then we have white ice, which tends to be kind of more opaque and doesn't let the light through as much. This is another kind of conceptual diagram from a paper from 2017, where we're looking at the light and the properties of it getting through snow. So you can see less light is getting through when there's snow cover on top of the lake. As soon as you have reductions in snow cover, you can get more light coming in. And this also has a kind of feedback mechanism where it keeps the phytoplankton up in those top surface layers. And so they're able to stay in the light for a longer period of time than they would otherwise with snow cover. And this is showing the chlorophyll A concentration from the top of the lake right under the ice cover down to the bottom. And you can see that the biomass sticks pretty well with those convective mixed layers. So we think this is also driving, how much is there and where it is in the water column. This is another example of the importance of snow depth. And so here I'm looking at all the different study systems and looking at snow depth on the x-axis and under ice photosynthetically active radiation, PAR or light on the y-axis, and it's a log scale. So the circles are when there was snow cover, the natural snow cover was there. And then the X's are where we shoveled off the snow, and then we measured the light again to see what the difference was. And we found that when we removed the snow, about 61% increase in the amount of light available to the phytoplankton, increased when we removed that snow. So significant increase in light just with the physical removal of snow, still ice cover present. And then we can use kind of thresholds for light limitation, and that's shown by that dotted line there, where anything over the line is considered to be, light sufficient. So enough light to be able to undergo photosynthesis. And so once we remove that snow, only about 5% of the samples were still light limited. So the majority, 95 of them, had enough light available to them. And so when we look at kind of projections of climate change in the future, any changes in winter precipitation or wind patterns and things that affect that snow depth are going to have big impacts on how much biomass and productivity is occurring under the ice. Here I'm comparing the two types of systems, both dimictic reservoirs, so these are the ones with ice cover up in the north, and then warm monomictic reservoirs, so down here in Missouri. On the left-hand y-axis, I'm showing this mean light in yellow, and then on the other y-axis, it's the chlorophyll A concentrations. And so going into this, I would expect that we would have the highest light and the highest chlorophyll A concentrations in the summer months. That's kind of one of our assumptions. And you can see that the chlorophyll A is highest in this dimictic reservoir in the summer, but you can see the light is in fact highest in the winter. And then down in the warm monomictic reservoir, both the chlorophyll A concentration is higher in the winter, and we see the light is not significantly different between these different seasons. So kind of counter to our assumptions, we don't see any, strong correlations between chlorophyll and light. And we definitely see these, non-differences or more light in the winter than we would have expected. Now I'll take you through the lakes from a northern perspective to a southern perspective. So starting with these Saskatchewan dimictic reservoirs, here we're looking at year-round chlorophyll A concentrations. And you can see that, it doesn't quite follow that bimodal pattern that we would expect to see. And if anything, the highest concentrations are occurring, kind of in those winter months. And so these green lines, again, indicate that kind of summer sampling season or, kind of traditional programs that the most of us employ. And we would miss those maximum concentrations if that was the only window that we were looking at these lakes through. Moving a bit farther south now into mesotrophic Lake Simcoe in Ontario. We can see here over multiple years, we do see kind of that bimodal pattern happening, but the peak in chlorophyll A concentrations is happening right before, kind of things open up and allow boats to go out and start the sampling season in the spring. So again, missing that maximum peak chlorophyll A concentration with our traditional sampling. Here in Missouri, we have a range of reservoirs in terms of trophic status, but we don't get ice cover very often. And you can see here over a 2-year period, we see this kind of, again, bimodal pattern. But again, those peak concentrations are happening outside of that winter. So we're seeing outside the window. So we're seeing the highest concentrations occurring in those shoulder seasons or non-summer months. Here in the town I live in, Columbia, Missouri, there was daily samples taken from a eutrophic urban reservoir. And here you can see over multiple years, we again see kind of this variability. This is again one reservoir, so lots of changes in chlorophyll A concentration. But again, those interesting maximums are happening outside of kind of the traditional sampling summer period. And so when we summarize this information by kind of normalizing these chlorophyll A concentrations, because they do represent a variety of different lakes, we can see the ones that have very little ice cover, so less than 70 or 70 to 100 days. Shown here, we see that the seasonal differences are in fact significant. So chlorophyll is higher in the winter months, in some cases 2 times higher than the summer concentrations. And again, I think this is because of that nutrient availability, where we have lots of loading and things happening in the winter months and lots of mixing. When we get less ice cover, more ice cover in the more winter months, we can see again maximum concentrations happening in the summer, but we still get those erratic peaks happening outside of that traditional season. So, much more differences that we see from month to month than we would have expected from this kind of traditional paradigm. So this is kind of a call for winter limnology or non-summer sampling, where there's a lot of really interesting stuff happening outside of that window. And I think now is the time to kind of get out there and start capturing it. And this may or may not be related to some of these news items we're seeing, where again we're hearing more about harmful algal blooms occurring in our inland water systems. We often warn the public, and our messaging is about what it looks like. So we often said, if it looks like this and it's relatively green in color, then don't swim in it. And so, based on the differences that we saw in, the chlorophyll A concentrations around the year, most of us for drinking water sources drink surface waters, and we do drink that year round as opposed to just in the summer. So are these kind of proxies or, assumption that green equals toxic relevant, and are they relevant year round? We know that the production of these toxins, cyanotoxins by cyanobacteria, have some pretty serious human and animal health risks. You can be exposed to it through ingestion and inhalation. So you don't even have to be in the water. You can actually get it through breathing these aerosolized toxins. So being in a kayak or taking a walk around the lake would still expose you if there was a bad bloom present. We can get it through the food web. So we know that it can get into fish and some of these other species that we eat and that wildlife eats. And then, of course, we can consume it in drinking water, which was the concern when there was an algal bloom in the western basin of Lake Erie for Toledo, Ohio residents. We can treat it to remove most of these toxins, but again, they have to be aware that a bloom is occurring and that they have high toxin concentrations, and then being able to do it at a large scale, which was, again, the issue in Toledo. So I'm learning as a relatively new academic you can't be everywhere all the time. And so how do you get year-round data, particularly for cyanotoxins? So this is not a sensor that you can put out to collect this type of information. But we can use volunteers and community or citizen science to be able to go out and get these year-round samples. So we have a high school citizen science program called ROS, or Reservoir Observer Student Scientists. This was a pilot project funded by the North Central Region Water Network, where we had high schoolers ranging in latitude from down here in Missouri all the way up to Minnesota and Michigan, collecting weekly water samples from their local reservoir or lake. And this is some work from some of the Missouri reservoirs. This is Rockbridge High School here in Columbia, Missouri, that collected all of these samples. Here I'm showing temperature groups, so the temperature of the water ranging from 0 to 15 degrees Celsius, 15 to 25, or 25 to 35. And we would expect that at the warmer temperatures, we'd have more chlorophyll and we'd have more cyanotoxins. And here we're looking at 2 different cyanotoxins. One is microcystin and the other is cylindrospermopsin. And so what hopefully you can see from this data is the surprising result that there wasn't significant differences between chlorophyll in the winter versus the summer. So that 0 to 15 degrees Celsius represents the winter months. There was higher microcystin in the summer months, as we would expect, but cylindrospermopsin showed the opposite trend, where we actually saw the maximum concentrations happening in the winter. Emily Kinzinger was my master's student who did this work, and she looked at factors that might be kind of stimulating or related to these off-season cyanohabs. The top 3 panels, A through C, are for microcystin, And the bottom three, D through F, are for cylindrospermopsin. She found that there was this significant positive relationship between water temperatures and microcystin, and also nitrogen concentrations. But for cylindrospermopsin, it was none of those factors. I mean, there was a relationship with temperature, but as you can see, it was kind of this U shape where the highest concentrations happened at the highest and coldest water temperatures. We also found a relationship with turbidity and light. So PIM represents particulate inorganic material. And we saw a positive relationship with cylindrospermopsin in that. And then again, kind of an inverted U relationship with light, where we had the highest concentrations kind of happening in those in-between light situations. So very different drivers of cyanotoxin production between microcystin and cylindrospermopsin. So this suggests that maybe there's this yin-yang relationship between these two toxins. And so even though microcystin seems to be highest when things are green, that doesn't seem to be the case with cylindrospermopsin. And so perhaps our kind of general indicators of toxicity are failing us. And we should be looking at other factors in addition to whether or not, the lake happens to be green or has high phytoplankton biomass. The other question that I thought maybe this audience would be interested in is understanding the productivity related to those chlorophyllite concentrations. So how productive are they? How much energy is that producing? And how will that facilitate things up through the food web up to the fisheries? And so here I'm showing the relationship between chlorophyllite concentrations under ice and AGP, or aerial gross production. So this is basically how much oxygen is produced by that chlorophyllae, or those phytoplankton. And you can see here that under ice, it still is a significant positive relationship. It can be rather noisy, but it does seem like some of that chlorophyll could translate to higher productivity, even under ice. And this is a comparison of both the production, again, AGP, with respiration, which is the AR, aerial respiration, the black bars here. And then, of course, the difference is the aerial net production. And showing the production rates for these Saskatchewan reservoirs, Blackstrap, Broderick, and Diefenbaker, relative to Lake Simcoe in Ontario. So you can see that the productivity under ice tends to be quite low, but we tend to get really kind of higher respiration rates. We often think of metabolism as this ratio of production to respiration, where if it's greater than 1, they're considered to be autotrophic, or production is higher, whereas it's less than 1, it's considered to be heterotrophic. And so we can see here that under ice, we're about half and half. So 2 of the reservoirs were dominated by respiration in the winter months, with P to R ratios less than 1. But both like Diefenbaker and Simcoe, kind of went over that balance and tended to look quite productive or autotrophic during the winter under ice. And then the second figure over here, the A through D, is showing the days since ice on in each of these systems. So A is Blackstrap, B is Broderick, C is Diefenbaker, and D is Simcoe. And we can see that this changes quite significantly in some, particularly on Lake Simcoe, as you can see, from days since ice on. And this is related to basically that snow cover and how much light is getting through that ice. And so at the end of the season, Lake Simcoe is a big windy lake, and it tends to have negligible snow left, and we can get some really high production rates occurring right before the ice comes off. And you can also notice the difference in duration of ice cover on these different systems. So when we put them all together and combine them, taking an average of this year-round metabolism, in the summer, we do find the systems to be autotrophic, so greater than that 1 in the P to R ratio. And in the winter, they're below the 1 when you average them all together, so tend to be looking like these heterotrophic factors. But again, the things that are influencing things in the winter are that light and ice cover and snow. And so if we see changes in those, we might start seeing a shift in this balance between autotrophy and heterotrophy. So to summarize, the question that we started with is what happens in lakes in the winter? We found that there was a sufficient quantity and quality of nutrients, both nitrogen and phosphorus. And this is most likely related in part to this internal phosphorus loading. So we know that a lot of that legacy phosphorus in particular is coming out of those bottom sediments and is available to phytoplankton to use. We also know that there's low under ice light due to snow cover. So the phytoplankton are light limited, but as soon as that snow cover is removed, they can respond quickly to increases in light. And so again, with predictions of climate change into the future, we might see some changes occurring at the, purely at the end of the winter when we have high windy conditions. There is under ice peaks in phytoplankton biomass, but not necessarily translating into productivity. We still see respiration dominating under the ice during those winter months. So although autotrophy can occur under ice, on a general basis, they are net heterotrophic. But the question becomes, what is the future going to look like if the baseline information? What can we expect when we have shorter winters? And what is the consequence of these climate-induced shorter winters? And so if you're a real pessimist, you could say, well, all our ice cover lakes will eventually lose their ice cover, like we've seen in Lake Zurich and other lakes in Europe. And they might be transitioning to what I see more often around here in Missouri, these warm monomictic conditions with no ice cover. And so there's the potential that this will create an environment in which cyanobacteria will win their competition with other phytoplankton, and we might have potential for more and different types of cyanotoxins. And so I think it's important to understand, and again, have this baseline information for winter and shoulder season dynamics, so we can kind of look to the future and make predictions about what lakes will look like on a year-round basis. And so to the question, does winter matter? I think it does, and that could be, shifting the scale. It could matter more in the future as we start to lose our ice cover and our winters start changing significantly. And so currently, most of our monitoring programs, we are missing these pieces of the puzzle of the fall and spring shoulder seasons in the winter, and perhaps it's time to start putting this together. So yes, as mentioned, all of you are starting to pack up your summer sampling gear. Well, maybe it's time to keep that out just a little bit longer and try to capture some of these off-season dynamics. Because these rapidly changing light and mixing dynamics could be facilitating the cyanobacteria growth and then allow for more cyanotoxin production. And again, this is maybe not as important on recreational lakes, but certainly for drinking water systems, this is going to have a lot of implications for human health and our ability to use these freshwater systems. So with that, I would like to acknowledge all the funders over the years. This is an accumulation of almost over a decade's worth of work. So lots of people to thank. And I'd also like to thank you for your attention. My email is there. I welcome any questions you might have. And I hope my internet's holding up enough to turn my video on and take your questions. Thank you.
Great. Well, thank you, Rebecca. That was really, really interesting stuff. So I will say on behalf of the 40 attendees that are muted, thank you. And I wish that we could have a live applause for you, but you'll have to accept my thanks. Okay, so for attendees, you have the option now to please submit your questions via the question and answer function. I don't see any questions in there yet. So, and I see from looking at the list of attendees, there are quite a few knowledgeable limnologists on here. So I know that you all are have some burning questions. I've been compiling a couple as we've been listening. So, so we will not be without questions, but now's your chance. Please submit your questions through the question and answer function if you have them. And again, a reminder that we will be sharing the recording of this video with all folks that registered for this webinar in a follow-up email. So I will get to the questions. And my first question for you is, you've made a really convincing pitch for us to be considering increasing the monitoring that we do in the shoulder seasons and during the winter. And so if I'm in the shoes of a water quality regulator or a lake monitoring program, a citizen science monitoring program, maybe I'm convinced and I want to start doing that, but I have budgetary limitations that sort of hinder my ability to go out and take daily samples, which wouldn't be maybe the ideal. So, so if you were thinking about the ideal time whereby a monitoring organization would collect data, maybe if they could only go out once an off season for each lake, or maybe 3 or 4 times an off season for each lake, how would you design that sampling program for nutrient and temperature and dissolved oxygen sampling?
Yeah, I think it, again, depends on what type of lake you had, but let's assume it's dimictic. You're going to want to capture those 4 different kind of mixing regimes. So, summer stratification, that should be easy enough. And then wait, as far as you can into the fall when you see, there's no difference in temperature from top to bottom. So you get that fall mixing because that's going to bring up those nutrients and could promote some algal growth. And then again, if you have ice cover, it's great to go out in the winter and get that sample through the ice if you can. And then again, when things mix in the spring and then the summer. So at least those 4 times, I think you can get a lot of information from. Logistically, I get it. It's really hard to get out all the time, especially when you're teaching and all the other activities we do. So yeah, we have some great collaborations. And as I mentioned, the Ontario government did it first to get water treatment plant operators. All they really have to do is fill a bottle. It's a pipe into the lake year round. It only represents that one area. But here in reservoirs, it actually is a little more representative because they're usually coming from the dam, which is the lacustrine or deepest part of the lake. So that's kind of a good, that's usually where we sample in the summer anyways. So yeah, I've got some collaborations here in Missouri. They seem pretty willing, we're sharing all the data and information with them. So mostly, they want to know for drinking water purposes. But yeah, so far it's been great partnerships just to get them to fill some bottles and put them in the freezer. And then they just ship them to our lab and we analyze them all here. So getting kind of, people that are already doing things on lakes without having to do the field investment is one way to go about it.
Yeah, great. Well, thank you. That's helpful. I know there are some citizen science monitoring coordinators and some water quality monitoring coordinators on this webinar. So food for thought there. Okay, we have some questions coming in, so I will go to the audience questions. Do you think zooplankton grazing may be temperature dependent as well and play a stronger role in summer than in winter?
Yeah, I think that's probably the piece that we know the least amount about is the zooplankton. And I've seen some more recent papers come out. I know Ted Ozerski just had a paper with some of his grad students kind of showing that, zooplankton are not doing nothing under the ice and that they're actually in higher abundance. I've seen it on my filters, you pull up a net or water and filter it, and there's a ton of very fat, happy looking zooplankton that are coming up in the winter. So I think, our assumption again was that their metabolism was limited by temperature. We often had that Q10 rule, for the doubling production with an increase in temperature, but we're finding that a lot more is happening under the ice in, zooplankton and even fish dynamics than we had previously thought.
The more we learn, the more complicated it gets.
Right. Yes, and the answer is never like, we're good, you can stop doing everything.
Okay, let's see. Another question is a fairly specific one. Have you seen winter or spring Planktothrix rubescens blooms in any of the volunteer monitored lakes? And I may have mispronounced that. I apologize.
Yeah, no, you got it. And I understand it. Yeah. So they tend to have a red pigment to them. I've actually got a great story where I got called because they thought there was a body under the ice. And it looks like blood coming up through the cracks in the ice. It's rather red pigments, and that's what it was a Planktothrix bloom. They can also kind of look like flowers, the formations of designs that they form under ice. So yes, I've seen those all the way from here in Missouri up onto Simcoe and some other Ontario lakes, those Planktothrix blooms under ice. So yeah, they definitely do well, and they produce toxins, unfortunately. So they're one of the ones that we always look for.
Gotcha. Interesting. I had never heard of those. So I'm a fish.
If anyone
guy,
calls you about, blood coming up from a lake, then that might be.
All right. Another question. Why do you think it is that internal phosphorus loading was higher in the winter in at least one of the lakes you looked at?
Yeah, so in that Lake Diefenbaker, it's fed by the tributary of the South Saskatchewan River. And so the flows are a lot less in the winter, which is, one of the new supplies of oxygen or what keeps things kind of moving through that system. So things are quieter and calmer in Lake Diefenbaker in the winter. And so, with that ice cover, the only new input of oxygen you get is from phytoplankton, producing oxygen through photosynthesis. And so we often get really low concentrations, winter fish kills are quite common in a lot of systems. And that's because it's gone anoxic under ice. And so that's promoting, the internal phosphorus loading, just like you would see in the summer months. So certainly ones that where flow has a big influence, and they tend to be higher in the winter in Lake Diefenbaker in that case.
Gotcha. Thanks. All right, we've got another one coming. For monitoring lake water when people aren't looking, have you considered monitoring systems? So there's a professor or a person at Purdue, Paris Collingsworth, who has some algae trackers just deployed in the Great Lakes. That might be an option for tracking phycocyanin and chlorophyll. And in regions where things don't freeze, that might work well. So have you considered any of those kinds of options?
Sure, I keep writing grant proposals to fund me for those toys, but yeah, no success yet. Yeah, I mean, that is a great resource. And, again, here where things don't freeze, you could use satellite imagery as well to get at some of those pigment concentrations. Now that's just the surface algae. And so we know in the winter, they tend to kind of peak right below the surface. So you might not capture it all, but it's more than we already know. So yes, definitely using those kind of sensor networks is the way forward.
Yeah. Yeah, and have you noticed any spatial patterns or distributions that you might be able to describe qualitatively in under ice sampling? I mean, is chlorophyll typically higher right over the deep hole in the lake, or is it more in the littoral zones, or have you noticed any patterns like that?
Yeah, so Lake Simcoe, we had the most kind of stations spread out over the lake. And it certainly was more related to ice cover and snow. So again, lakes freeze from the outside in and vice versa. So it was wherever that clearest black ice was forming. So where you got kind of less, when it freezes and thaws, that's when you get that opaque white ice. So wherever the black ice was for, whatever reason, and then certainly the lakes that were more exposed, the one Lake Blackstrap was kind of in the bottom of an embayment. And so it had a lot of protection from the wind. And so it accumulated a lot of snow. So there was very low production rates in that lake. Whereas the wind exposed, ones like Lake Simcoe and stuff tend to have higher rates. So yeah, wind, anything that would relate to snow cover and types of ice would have the biggest influence.
Gotcha. Thanks. Okay, I've got some more questions here. Let's see. How important is it, do you think, to also try to monitor upstream in the winter to try to get a better sense for the nutrients coming in from, whether it's a stream or a tile drain coming off of an ag field, those sources?
Yeah, I mean, they can be important. That's, why we're kind of pushing these BMPs or best management practices to keep, kind of crops and winter crops and things like that on the land year round, so that we don't get those large pulses coming off, particularly in the winter. Because yeah, if they have enough light, those phytoplankton are responsive, and they, if they need the nutrients, they'll be able to take them up. So yeah, it's not unimportant, but I'm sure, again, logistically difficult.
Yeah, I think that's right, especially manure spread over frozen ground doesn't absorb well and flows right off sometimes. We've got a couple questions that I think we can lump together, and we talked about it a little bit already, but maybe we can go into a little more detail. What parameters would you recommend that be monitored, and do you have any frequencies recommended for specific parameters to monitor in the shoulder and winter seasons?
Yeah, I mean, if you can get it weekly, great. But once a month, you should be able to capture kind of all those different patterns at least. And kind of our classic nutrients, and chlorophyll. The toxin was kind of interesting. We just kind of tagged it on because it was something my lab was regularly doing, and I was surprised at how high the toxin concentrations were. And those are easy. That's just water that you freeze. So that's not a hard one. So yeah, I would say if you can get some chlorophyll and some oxygen, if you had the meters or some kind of sensor or probe, you can get at metabolism that way too. It's kind of, we call a free water metabolism. So looking at oxygen concentration changes with sensors.
Gotcha. Thank you. Okay, here's another one. So I know here in Michigan and probably throughout the upper Midwest, in some places there are aeration systems that are operating, and those can operate in small spatial setups. They're protecting a dock or maybe a harbor, an entrance to a harbor. So some critical infrastructure. In some cases, those aeration systems are operating more in the center of the lake and might be really moving water and keeping the lake more ice free. So I don't know that you've presented any data on that, but you I think have some tools with which to hypothesize on how might those sorts of systems affect the winter productivity within a lake?
Yeah, so let's assume that they work properly and and they're pulling oxygen in and mixing the whole water column right from top to bottom. They would definitely help with some of the oxygen, issues like fish kills and things like that. They might even prevent, internal phosphorus loading from occurring a lot in the winter because of that oxygen. But they also create a situation where cyanobacteria win in a competition with other phytoplankton. So they have like little balloons inside them and they can move up and down in the water column, whereas some other ones that are the healthy, good phytoplankton like diatoms that we want to have around, they can't move. And so they would be disadvantaged by being circulated, that deep into the water column consistently, whereas cyanobacteria may win and dominate because they can stay up high and get enough light. So yeah, it depends whether you want to, have a good fisheries or drinking water. Depends on the use of the water.
Yeah, that's a tough
It's always our
choice.
challenge. Yeah.
Yeah, and maybe not mutually exclusive. Cyanobacteria
Yeah.
can hurt the fisheries as well. So
Exactly.
okay, well, thank you. I believe I am seeing all of our questions answered. So I will give folks one last chance to get questions in. If you have any questions, let's see. Yep. So seeing no additional questions, I think at this point we will. Oh, we got one. So we often tell lake residents and recreationists that blue-green algae prefer warm, still water when trying to explain when and why they occur. But with the uptick of winter blooms in our area, this is kind of confusing for our residents. Do you have any advice for how to communicate these issues?
Yeah, I mean, it depends on the type of cyanobacteria. So there's different types of cyanobacteria that produce different types of toxins. And so, yes, some of them, definitely do better under those situations of calm and warmth. But others, like we just talked about, that Planktothrix tends to do better. It can't fix atmospheric nitrogen and it has different needs than different types of cyanobacteria and produces different types of toxins. So yeah, sometimes that is true, but in the winter you can get different types of cyanobacteria that do better under low light conditions, but they also produce toxins. Sorry, I know there's no clear message on this one.
It's complicated.
It's complicated.
Yeah. Well, I think that makes sense. And Adrienne, who asked the question, said thank you. So I think it's been helpful to her. Certainly helpful for my understanding. Okay, I think I see no more questions. So at this point, I will again say thank you on behalf of the MGLP and those folks that attended. This has really been an interesting talk and it's gotten me thinking a lot as a fisheries biologist about when to do a little bit more monitoring on the lakes that I'm interested in for some of our more sensitive species. So thank you. And again, folks, we'll be sending out an email with the link to the talk that you can watch or share with folks that you think might be interested. And you can sign up for future MGLP webinars on our website. So thank you, everyone, and have a nice day.
Thank you.